Hi, I'm Monique Weldon, and I'll be talking about the molecular basis of cancer and what I've learned during my 8 weeks in this course. I can't wait to tell you about all the things I've learned while reading for this course. There were a lot of concepts we learned during this course, and I could cover a lot of them, but I will only focus on a few that really stood out to me. I will discuss cancer as a genetic disorder briefly, then infectious agents and oncogenesis, the tumor microenvironment, tumor angiogenesis, biomarkers for cancers, interferons, and colorectal cancer, as it was the main oncological disorder I investigated this summer. I included this section as something I had learned during the course because this was something I had never thought about. I had never thought about how people were able to know that cancer was based on alteration of the genome in cells. I thought this was an interesting way to start the course and an interesting way to start the presentation. In the 70s, Bruce Ames found that there was a correlation between the mutagenic potencies of various compounds and chemicals and their abilities to induce tumors. He found that carcinogens were able to damage cells and the DNA of cells. This was a novel approach to the discovery of the development of cancer. Prior to this, there had been several theories on the development of cancer, but none had stood up. Much later, scientists discovered cancer-causing genes, also known as oncogenes. This development was based on studies of the SRC genes in RSV. Proto-oncogenes potentially become activated into a cancer-causing oncogene. Cancer cells can carry mutant cancer-inducing genes in their genomes, which can influence surrounding cells in their genomes to promote tumor differentiation. Tumor formation does not happen all at once, but is instead a multi-step process. Single oncogenes cannot by themselves change normal cells into cancer cells. However, multiple oncogenes can cause tumorigenesis. Growth normalizing genes, aptly named tumor suppressor genes, were found to restore the cell to normal functions and behaviors. Here's just a little snippet of other things that we learned in this topic. The most commonly mutated gene in all cancers is TP53, which produces a protein that suppresses the growth of tumors. Inherited mutations in the BRCA1 and BRCA2 genes are associated with hereditary breast and ovarian cancers. Before taking this course, I actually had no idea that infectious agents could cause cancer. I had never really thought about what other causes of cancer could be besides UV rays from the sun and genetics. When I thought about things that cause cancer, those were the only things that came to mind. Even after I had taken the Gardasil vaccines against HPV when I was a teenager, I had still not considered that an infection could cause cancer. It is interesting now because I have recently seen commercials that encourage HPV vaccinations, because later on, cancer can form from these infections if a person is not vaccinated in their teens. I guess this means that awareness of potential cancer-causing infections is not well known outside of the scientific community. It was once thought that infectious agents did not contribute to the development of cancer. Since we can now connect infectious agents to cancer, it has given us a way to try to develop vaccines to prevent cancers, such as the aforementioned Gardasil vaccine I received. One of the first mentions of cancer due to infections was in 1911 when Roos found that viruses extracted from a tumor-laden chicken could be injected into another chicken and yield the same cancerous results. There are 3 ways that infectious agents can contribute to cancer and DNA damage to cells. The first way they can contribute is through causing persistent infections with increasing in the amount of metabolic and reactive oxygen in a cell. As we've reviewed in this course, reactive oxygen can cause damage to the DNA via mechanisms such as hydrogen atom abstraction in the methyl group decomposition of cytosine derivatives. The second way these agents can contribute to cancer is through their activation of proto-oncogenes to oncogenes, or their deactivation and suppression of tumor suppressor genes. The third way infectious agents can contribute to human cancers is that they can inactivate or suppress the immune response, allowing cancerous or mutated cells to thrive. We now know that there are infectious agents that can cause cancers, though there usually needs to be other factors or cofactors to the infectious agents in order for there to be cancer in a human. Infectious agents that can contribute to cancer development include viruses, bacteria, and parasites. Viruses alone will not cause cancer in humans, but these infectious agents can be the precursors involved in the early steps of cancerogenesis. One of the infectious agents I learned about in this course was Helicobacter pylori, which can contribute to gastric cancer and mucosa-associated lymphoid tissue, or MALT. H. pylori can produce oxidative free radicals, which can cause DNA destruction and damage, which could result in oncogenesis. Another way H. pylori can potentially affect the cell would be its production of the protein CAG-A, which can activate SHP2, a tyrosine phosphatase. Activation of SHP2 interrupts cell-to-cell adhesions and junctions, which can affect whether cells move around or stay where they are supposed to be. These cells could be implicated in later metastasis. Another theory on how H. pylori causes oncogenesis is that it produces the protein FAK-A, which in turn prevents immune cells from effectively killing the bacteria. H. pylori has also been investigated as being a contributor to chronic inflammation in cells, which migrate and lead to metastasis. Before taking this course, I had not considered the microenvironment of tumors and how that would affect tumor development and proliferation. From this course, I learned that the tumor microenvironment consists of cellular components of the tumor, the surrounding extracellular matrix, fibroblasts, endothelial cells, immune cells, and interstitial fluid. Tumors and their microenvironments are a delicate balance of signaling and proliferation. There are ways to either promote or suppress the microenvironments of tumors. Wounding promotes the formation of tumors. When only exposed to a certain carcinogen, such as benzoprine or UV rays, there is little tumor development. But when the doses of the exposure are high enough to physically damage the tissue, or there are wounds already present in the tissue, tumors will readily grow. Tissue can be readied for tumor growth and development from chronic irritations or prior injuries, such as chemical burns or sunburns. This was interesting to learn because I did not know about the correlation to childhood sunburns and the later development of skin cancer. So not only did I learn information that would help me on the tests and papers for this course, but this is also applicable to real-life situations. Embryonic microenvironments can induce tumors to grow and function as if they were normal cells. Embryos are in a microenvironment that somehow allows for the override of tumor-promoting oncogenes and the suppression of tumors. In embryos injected with malignant tumor cells, extracellular regulatory cues in the microenvironment of the cells help to signal the tumor cells to suppress their tumorigenic phenotype. Tumors can be conceptually thought of as wounded organs that do not heal. When an organ is injured, a cascade of events take place to restore functions. Molecules such as tissue factor, fibrin, and MMPs are called to the scene of the injury to induce remediation of the wound. MMPs are related to unregulated tissue growth, tissue invasion, and metastasis, and can be caused by altered extracellular proteolysis. A cascade of events follows injury, which promotes organ repair but also reduces adhesiveness of the epithelial cells, increases circulating cell types, increases cell proliferation, and cellular migration. In a normal context, epithelium differentiates. Activated cells do not persist, and stimulatory growth factors and cytokine concentrations decrease in the microenvironment. In the tumor microenvironment, however, overexpression of a single extracellular matrix enzyme can effectively manifest in all the signs of tumor formation. MMP overexpression can cause a host of problems, leading to reactive stroma formation, such as expression of extracellular molecules. There are extracellular matrix molecules that are usually only associated with wounding, oxidative damages, and genomic instability. Another aspect of cancer formation that I did not know about before taking this course is the development of vasculature for the tumor that supports tumor growth. Tumors more than 2 millimeters need to have some sort of vasculature in order to function. Normal blood vessel formation is highly organized and structured compared to tumor vessel formation, which is disorganized and misshapen. The vessels associated with tumors often end in open-ended pool lakes, which is thought to be caused by dysregulation of angiogenic signaling in the tumor bed. In tumor angiogenesis, as in normal development of vessels, there is budding of endothelial sprouts and bridge formation. However, in tumor cells, there is uneven blood flow and localized hypoxia due to the aberrant formation of the vasculature. There is increased permeability in tumor vessels due to disrupted tumor endothelial cells and reduced coverage by parasites. Endothelial progenitor cells contribute to tumor vessel formation by differentiating to endothelial-like cells and promoting the expansion of other endothelial cells. Bone marrow-derived myeloid cells contribute to tumor vasculature by expressing cell surface markers such as Ti2 and CD11b. Due to the nature of vasculature in tumor environments, there are severely hypoxic areas, which are often composed of dead or dying tumor cells. The hypoxic nature of tumor environments protects tumor cells against radiation therapy, which relies on reactive oxygen intermediates. Tumor proangiogenic factors can include VEGF, angiopoietins, and others. VEGF binds to the receptor VEGF receptor 2, or FOLK1, which will activate PI3K, SRC, and protein kinase C pathways, leading to proliferation and gene expression in endothelial cells. There are several pro-angiogenic factors in tumor development. VEGF is the most frequently targeted for tumor angiogenesis in human patients. VEGF has a prominent role in tumor angiogenesis, making it a profitable target for tumor therapies. VEGF antibodies are used to seize VEGF or block VEGF receptors and VEGF-dependent signaling. The most well-known VEGF antibody that is currently past clinical trial stage and is being used in humans is Avastin, which blocks angiogenic activity. Before taking this class, I did not even know what biomarkers were. I had never heard of the term before. But now I am very familiar with biomarkers and their clinical relevancy. Biomarkers are cellular, biochemical, or molecular changes in a biological system which are helpful for indications of cancers. Biomarkers are divided into 3 types: biomarkers of exposure, biomarkers of effect, and biomarkers of susceptibility. Biomarkers of exposure indicate the presence of a carcinogenic substance or its biological interactions within cellular molecules. They provide the most effective direct verification that a human has been exposed to a carcinogen. They are the actual chemicals or metabolites that can be measured in the body or from bodily excrements to determine the characteristics of exposure. Biomarkers of effect refers to the response of the body to exposure to a carcinogen. They can indicate early subclinical changes that could potentially have pathological consequences. A good biomarker of effect needs a robust measure in response to the carcinogen and should be minimally affected by inter-individual variability. An effective biomarker of effect is chromosome aberrations in peripheral blood lymphocytes, which have a positive correlation with increased cancer risk. Chromosome translocation is widely studied and is the most well-established biomarker of exposure and effect, as it has been seen in almost every cancer type. Cancer can be caused by a single translocation of a chromosome, such as the case in chronic myelogenous leukemia caused by a single Philadelphia translocation. Biomarkers of susceptibility can help with risk prediction and can help identify high-risk individuals with an increased sensitivity of target molecules in critical times when exposures can be most detrimental. Biomarkers of susceptibility indicate the natural characteristics of humans that make them more susceptible to the effects of exposure to a carcinogen. Interferons are cytokines that are modulators of the immune response in response to viruses, bacteria, and parasites. Interferons can inhibit cell proliferation and can either inhibit or promote cell differentiation. Interferons are responsible for transcription of genes that produce a variety of cellular responses, such as promotion of natural killer cell activity, increase of antigen presentation and lysosome activity of macrophages, increased normal cell expression of class 1 major histocompatibility complex molecules and class 2 MHCs on antigen-presenting cells. While interferons are widely regarded as inhibitors of cell proliferation, recent research has shown that they can play a role in oncogenesis. One example of interferon inactivation contributing to cancer is the case of deficiency of interferon gamma or its receptor, promoting colorectal cancer development. Other studies suggest that interferon gamma treatment has both anti- and pro-oncogenic activity dependent on the cellular, microenvironmental, and molecular context. Toll-like receptor signaling is important to oncogenesis for its gain-of-function mutation in adaptor protein MYD88 for toll-like receptor signaling in MALT. Interferons mostly contribute to oncogenesis in a tumor-suppressive manner. They can do this by activating cascades to promote apoptosis of tumor cells, increase the cellular immune response, and can inhibit metastasis of tumor cells. Interferons push cells towards apoptosis, though it is in a different manner than the quick process of cytotoxics via camptothecin. Interferons induce apoptosis and angiogenesis through signaling interferon alpha receptors. The organ I was assigned for this course was the colon, and I learned a lot about colorectal cancer while in this course. I was able to put all the information I learned in the course together to really thoroughly write about and understand colorectal cancer in the last paper. Here I will tell you a little about what I learned about this cancer. Colorectal cancer is the third most common cancer type in the United States, and is also the third leading cause of cancer-related deaths in the U.S. Colorectal adenocarcinomas comprise about 95% of all colorectal cancers. Worldwide, colorectal cancer accounts for about 9% of all cancers and is the third most common cancer in the world. Colorectal cancer equally affects both men and women. Interestingly, more developed countries such as the United States, Western Europe, and Australia have higher incidences of this cancer compared to less developed parts of the world, such as countries in India, Africa, and South America. In most cases of gene mutation caused colorectal cancer, there are only 2 mutations needed to initiate colorectal tumorigenesis. These mutations usually occur at the adenomatosis polyposis coli, tumor suppressor locus 5q21 and 5q22, which leads to the shortening of the adenomatosis polyposis coli protein, which has been detected in over 75% of sporadic colorectal cancers. The APC gene mutations alone are not sufficient enough to be the sole cause of initiation of carcinogens. More than one gene is involved in the progression of an adenoma to colorectal cancer. In these adenomas that become malignant, the KRAS, NRAS, and HRAS genes that encode for the small GTP binding proteins are activated by point mutations and prevent the activation of GTPase. Chromosomal instability through loss of heterozygosity at loci in APC, T53, and SMAD-D is found in 80% of sporadic colorectal cancers. It is recommended in the United States that all persons over the age of 50 with risk factors for cancer be screened for colorectal cancer. I mentioned Avastin when I talked about learning about tumor angiogenesis. Avastin, in conjunction with standard chemotherapy, is now the first line of treatment given to patients with metastatic colorectal cancer. I would have to say I learned a lot in this course over these past 8 weeks. I've never thought of cancer more than in this course, and I really think I have a much better grasp of the concepts related to cancer and especially to colorectal cancer. I have learned more about the role of proteins, genes, and the growth and promotion of cancer than I knew before. I will use this understanding of the concepts of cancer mechanisms in future courses. I'm excited to learn more about the development of treatments for cancers, and I'm excited that I will be able to better understand the science behind these developments. This is my reference list for this presentation. Thank you very much for listening. Have a great night.